I have you loud and clear.
Hello. Welcome. Science.
And that is to say,
physics,
medicine,
nature,
or
Space. Time.
the brain,
Life. The universe.
Hello, welcome to The Naked Scientist, the show where we bring you the latest breakthroughs in science, technology and medicine. And that's with me, Chris Smith, and with Phil Sansom.
This week, we delve into the disease that accounts for a quarter of all cancers in men, prostate cancer. We'll be finding out how the condition is picked up and diagnosed.
Plus, a way to cut aeroplane emissions by flying just a bit higher. And were dinosaurs hot or cold-blooded? How scientists can now tell a dinosaur's temperature just from its eggs.
The Naked Scientist podcast is powered by ukfast.co.uk.
The new coronavirus from China has continued to dominate the headlines this week. On Thursday, the initial 83 people evacuated from Wuhan City and quarantined near Liverpool were confirmed disease-free and released. I spoke to one of them about his experience.
My name is Khan Lambert. I'm a PE teacher living in Wuhan, China, and I'm currently in quarantine on the Wirral. We were evacuated by the British government a few weeks ago.
Have you seen much of the action when you were in Wuhan?
Yes, there was a lot of activity going on while I was there. People being taken to hospital, a lot of overcrowding in hospitals, people fighting to see doctors, people fighting to get food when the foods were running low.
Were you scared?
On a personal level, I wasn't scared. My biggest worry was my 80-year-old grandmother who suffers with COPD and was visiting me in Wuhan and got caught up in all of this.
And what was it like when you actually got evacuated? How did that play out?
It was chaos. We got a phone call about an hour and a half before we had to be at the airport. And I mean, we wasn't fully prepared because we wasn't expecting to hear until the following day. We were waiting around for 4 or 5 hours in the cold at the meeting point because they kept pushing it back. And then once we got inside the airport, chaos. I mean, there was no real organization. Flight tickets were blank with just a simple number on. Nobody really knew what they were doing until we got on the flight, actually. And then once we were on the flight, the staff were fantastic with us.
And when you touched down, apart from the enormous sense of, I finally made it home, what happened next?
There was a massive sense of relief once we touched down in Brize Norton. But then we were obviously ferried onto buses and transported with police escorts up to the Wirral.
And how are they keeping an eye on you? I don't mean as in policing that you're there. I mean as in from a health point of view.
At the very beginning, they were doing checks 3 times a day that we felt okay, that we weren't showing any signs or symptoms. They didn't actually test us at any point because I think the advice from Public Health England was that it is pointless testing anybody that's not showing any signs or symptoms because it just wouldn't show on the test. They have since changed that advice, and so we were tested for the first time yesterday, and we're just waiting for those results to come through.
And what about your future? Because if you work in Wuhan, are you on the next plane back out there?
No, I'm not on the next flight. Obviously, the airport in Wuhan is closed. I can't get back into Wuhan now until these restrictions that the Chinese government have put on Wuhan are lifted. So I don't expect to go back to China for another 4 or 5 months, maybe even 6 months.
What does that mean for your job then?
Thankfully, I work for a very reputable school in Wuhan. So my job is safe, and all of the expats that work there, their jobs are safe.
Is the school still open, or is basically everything off?
No, school is actually closed, but they did start online classes yesterday.
You're not going to do some exercise classes online then?
No, it's a little bit difficult to do that. I think I've got a bit of a lucky break on that respect.
Wuhan City PE teacher and ex-coronavirus quarantinee Carl Lambert. One of the other major developments this week was a sudden apparent surge in the numbers of people reported by China to have the infection. In one day, the rate of diagnoses increased almost tenfold. This, China explained, was because they'd altered the way they were diagnosing the disease. So what might be the implications of these much bigger numbers of cases?
My name's Vera Jeffcott and I'm a research fellow at the University of Cambridge where I study outbreaks of unknown etiology. Obviously, the size of the outbreak has grown significantly, and that's because of how transmissible this virus looks to be, even with some of the quite extreme control measures that have been put in place in China. I think what's interesting, though, is whilst the numbers have increased steadily within China, we're still only seeing limited cases in other countries.
Do you think that's real, or do you think it's a symptom of the fact that when you go looking, you find, and if you don't look, you don't get the answer to a question you don't want to ask?
Oh, I think that's the big question here. I would say, considering what we know about the virus, and actually a lot of what we don't know about the virus, such as when people are infectious, if it's before they've developed symptoms, if it's after, I would think that probably there is some unobserved transmission going on in some other countries.
Now, China this week have changed how they define a case. Does that tell us anything about the real numbers that are probably happening in China, and therefore anything about what's probably happening elsewhere around the world?
This one is quite interesting because obviously now that it's a broader case definition, we're going to be capturing a lot of other cases that previously were being missed. It's unclear if the reason that testing wasn't a good enough proxy by itself is because the testing was giving negatives when it shouldn't have, or there weren't enough tests, or they could only run so many a day. But this looks like a reasonable way now of getting a broader sense of it, and potentially also capturing some of the milder cases too that might have been going under the radar previously, but will actually give us a better sense of actually how many people do develop severe illness from this and how many go on to die, which could be smaller than was initially thought.
And given these very big numbers being reported in China, one therefore must presume that there were big numbers previous to now as well. What does that say about the likelihood of it already being in countries like this one?
Well, so it's unclear because they did take quite strong lockdown measures quite early on, but it does mean that probably some people were acting as if they thought they were negative and perhaps interacting closely with friends and family anyway, and including outside of the areas that were locked down, which could have well led to more of this unobserved transmission to other countries.
Freya Jeffcott. Also this week, scientists were racing to file bids for funding announced as part of an emergency response programme to bankroll the development of vaccines against the new coronavirus. One of the applicants was Cambridge University's Jonathan Heaney. His team have developed a system to enable them to produce vaccines based on the genetic material of the virus.
Coronaviruses are named after what they look like. Round spherical objects with these little, what we call spike proteins.
Those spikes are how the virus docks with and invades our cells, so an effective vaccine has to produce antibodies that can block them. But that's actually easier said than done.
Because if you make antibodies to the spike, actually those antibodies can help drag this virus into monocyte macrophages, a very important inflammatory cell. It can be reprogrammed by these viruses, making the disease much more severe than it would be.
So an ill-conceived vaccine could potentially pour fuel on the microbial fire and accelerate the infection, paradoxically making the person even more ill than if they'd never been vaccinated.
So the vaccine strategy really has to be laser-specific, targeting those domains that actually are absolutely critical for docking with a cell. That's this new technology that we've developed.
What their technique allows them to do is to screen, using a computer model of the virus structure, all of the components of the viral coat, including those spikes that it uses to get into our cells, to find the crucial pieces that will work as a vaccine and disable the virus, but without the risk of making the infection worse.
This is a computational approach where we take the genetics of these viruses and we target those essential elements in a laser-guided missile type of way so that the antibodies attach only to those regions that is important for docking.
The result is a piece of the viral genetic code that the virus uses to produce that part of its coat. This genetic sequence can be injected into the body where immune cells pick it up, decode it, and then use the information to educate the rest of the immune system to produce antibodies against that discrete part of the virus. But how long, crucially, is this going to take?
It has to then legally be manufactured in such a way that the regulatory authorities will allow it to go into humans. It needs to go for separate safety and toxicity testing. That will take 4 months.
Is this going to arrive on the scenes fast enough to make a dent in this outbreak?
I think so. I think the cat is out of the bag, meaning the virus is out of China. The Chinese are having great difficulty containing it. This virus has changed the rule book. People actually who are not overtly ill, sometimes don't even have fever yet, can be contagious. So it's a different game. We do need to move quickly, but the golden rule must be do no harm, meaning don't be in a rush to make something that's going to make the disease more severe.
Jonathan Heaney, who's hoping to produce a vaccine against the new coronavirus.
UK scientists have discovered a way to cut the airline industry's contribution to global warming by as much as a third. The answer lies in minimising the contrails that form when a plane flies through a humid patch of sky. These contrail clouds reflect heat heading for space back down towards the planet's surface, forcing up the temperature.
Now, a new technique is enabling scientists to spot which flights are the worst offenders and how they can avoid the problem. Megan McGregor's been hearing how from Imperial College's Mark Stetler.
What we were able to do is use a high-resolution data set of aircraft trajectories in the Japanese airspace. And what that showed us was that 2% of the flights contributed to 80% of the climate forcing. So it's a really skewed distribution where you have a very small number of flights contributing to the vast majority of the warming effect.
And how does this contrail cloud cause warming?
There's a balance between two processes. The contrail, if it persists and spreads, it will both reflect incoming sunlight back out to space, and that's a cooling effect, but it also trap outgoing heat that would otherwise go out to space, and that's a warming effect. Now, during the day, there's sunlight, and so the cooling effect and the warming effect balance each other out. But then overnight, if a contrail persists, there's no sunlight, so there's no cooling effect, and so what we're left with is a warming effect. So it's really the ones that persist for a long period of time into the nighttime that have the warming effect.
If you wanted to take a targeted action towards this sort of small number of flights to reduce their contrail formation impact, what would it be that you could do?
Because contrails need this humidity in the atmosphere, actually there's regions of the atmosphere that are humid enough, are relatively thin layers of the atmosphere. And so small altitude changes, either up or down by 2,000 feet, which is what we simulated in our study, would mean that an aircraft could fly outside of that contrail forming region. So an aircraft that originally had formed a contrail, if we just change the altitude by plus or minus 2,000 feet, may end up not forming a contrail.
And that sort of is a different approach to mitigating, for example, carbon dioxide emission.
The plane is still going to emit carbon dioxide. What we were careful to look at was whether the changes that we were making to the altitude of flight would have a significant effect on the fuel consumption of the aircraft, because CO2 scales with fuel consumption. And that effect of CO2 that we put into the atmosphere will persist for a few hundred years. Compare that with a contrail, which lasts for less than a day. If we only made changes to the altitude of flights, this effect of contrails is something that we could almost switch off, get rid of that effect on the climate system from aviation.
If these sort of high humidity areas are not that difficult to avoid, why is it that that's not part of the sort of route planning now?
In the process of flight planning, you have to rely on forecasted weather data. And so one of the pieces of work that we're looking at now is how accurate those weather forecasts are in terms of predicting where there's that high levels of humidity for contrails to form. What's encouraging is that pilots and air traffic controllers already have a discussion while a flight is ongoing, so en route. Adding contrails to that discussion would be another dimension, complicating the calculation slightly, but it's something that we could incorporate. But we are working towards that, and we're looking at engaging with air traffic management service providers.
An interesting discovery, and they'd be plain daft not to use it, Shirley. Mark Stetler there discussing work that has just been published in the journal Environmental Science and Technology.
Hello, sorry to butt in. Katie here from the Naked Scientists. Did you know we make other naked shows too?
The fraction of all humanity who has actually gotten a chance to see their own brain is very tiny, and you are welcomed to that club.
So if you enjoy musing over the mind, reflecting on thought, or frankly feel bamboozled by the brain, check out Naked Neuroscience.
Well, my face hurts now, so yeah, let's go. It's spicy.
Don't go down into the creepy cellar and turn the light on.
Exactly.
Access the full archive via nakedscientist.com/neuroscience or subscribe to Naked Neuroscience wherever you get your podcasts.
On the way, new powers for the UK to regulate social media and our in-depth look into prostate cancer.
Now, from very high altitudes to very high energies, scientists have unlocked the next stage in the cutting edge that is experimental physics. This is an upgrade to facilities like the Large Hadron Collider, where beams of tiny particles race round at nearly the speed of light and then smash into each other to reveal what they're made of. The results shed light on the fundamental nature of the universe. The experimenter's ideal next step, though, is to up the ante using muons. These are essentially heavy electrons, and which can be collided at even higher energies. But these have been very hard to make into a focused beam.
Now, an international collaboration has managed to create that beam in what they call the muon ionization cooling experiment, or MICE. I got some concise MICE advice from research leader Chris Rogers.
We've demonstrated a technique whereby we can take a beam of particles called muons and we can squeeze them right down and accelerate them up to really high energies. Because of the unique properties of muons, we can actually explore physics which is even beyond the energy scale which is available in the Large Hadron Collider.
Wow, this is real futuristic.
Yeah, right, that's it. And no one's developed a technique like this before which can really be used to handle muon beams.
What exactly is a muon to start with?
So a muon is like a really heavy electron. You actually have muons going through you pretty much every second of every day, which come from cosmic rays.
I have a muon in me right now.
That's pretty much right.
What do they look like?
Just like electrons, except for a couple of special properties. One is that they're much heavier than electrons, almost 200 times heavier than electrons. And the other one is that they decay radioactively, so they only live for 2/millionths of a second.
That's bizarre. How do you even deal with them?
We have a special trick up our sleeve. If you accelerate particles to really high speeds, as the particles get closer and closer to the speed of light, they live for longer and longer. It's Einstein's time dilation phenomenon.
How do you make the muons in your lab?
We take a beam of protons, accelerate those protons, and then bash them into a target. All sorts of other particles come out, and some of those particles are muons.
And how have you been trying to deal with them in this particular experiment? Because this is the first time you've managed to get them into a beam, correct?
We've had muons in a beam before, but we've never really managed to prepare a beam so that it would be suitable to accelerate them. Much more like a laser beam, if you like. We pass that muon beam through an absorbing medium, and as the muons go through the absorber, they lose energy. All of that hot gas slows down as it goes through the absorber. So then we need to accelerate that beam back up using a conventional particle accelerator technique.
What's this material you're filtering them through? Is it something special and strange?
We use either liquid hydrogen, cooled down to a few tens of Kelvin, or we use lithium metal with hydrogen embedded into the metal.
Those are strange and weird.
They're pretty cool bits of kit which we use to do it.
Why those?
When the muons go through the absorber, they knock the electrons off the atoms. And when we knock the electrons off, the muons lose energy. That's what's called ionization. And that's why the technique is called ionization cooling. Now, there's another thing which happens. They bash into the center of the nucleus and they scatter off, flying off in all sorts of different directions. Now, we don't want that. So we have to pick special materials where the nucleus of the atom is as small as it possibly can be. Hydrogen has the smallest nucleus of any material, and lithium has a pretty small nucleus as well.
Is that why it's taken so long to figure this out? Because you're trying to get materials with small enough nuclei? Nucleuses?
Nuclei. It's not just the material which we have to consider. We have to combine that with a particle accelerator lattice. And combining those two different things into one experiment was really tricky.
What did it feel like when you finally managed it for the first time?
It was pretty cool. In fact, we only cooled the beam by about 10% of the full cooling channel which you would need in a real muon collider facility. But that was pretty cool.
And what does this mean for physics? Is there really exciting science coming up that could potentially use muon beams?
The aim of our experiment then is to take this technique and then put it into a thing called a muon collider, where we collide beams of muons together. Muon colliders are really exciting because they let us reach much higher energies than are available using even existing facilities like the Large Hadron Collider at CERN.
It's like a Large Hadron Collider upgrade.
It would be a Large Muon Collider.
Large Muon Collider. That's cool.
Should be.
We'll have to wait and see what happens when they try it. Chris Rogers there from the Rutherford Appleton Laboratory, discussing the experiment they call mice and which was published in the journal Nature.
And now we're heading back in time because Adam Murphy's been looking at some cracking, cutting-edge science using eggshells to take a dinosaur's temperature.
Jurassic Park is one of my favourite films. I watch it several times a year, but as the years go on, the picture on screen gets further and further from reality, and the scary lizard-like thing peering in the window of a Jeep probably didn't match what dinosaurs look like, as dinosaur expert Robin Dawson was quick to point out to me.
Well, I think for as long as we've named dinosaurs, we've gone from thinking they're lizard-like and reptile-like to now, over the years, learning the group of dinosaurs, which includes the scary velociraptor that's in all the movies. This group of dinosaurs that birds evolved from, the more we learn about them, the more characters they seem to have that are kind of bird-like.
We know T-Rex had feathers, for example. We also know the T-Rex was warm-blooded like a bird, not cold-blooded like a crocodile. But Robin's been working on a different technique to measure dino temperature by looking at the eggshells of dinosaurs. Eggshells contain carbon and they contain oxygen, and sometimes these elements will have an extra particle called a neutron inside them, and that makes them heavier. If a heavy carbon binds with a heavy oxygen, it makes a much stronger bond, and it takes more heat to pull them apart. So if you look at the eggshell and it's got none of those kinds of bonds, that means that when Mama Dinosaur was making the eggs, she must have been running hot enough to stop them forming. And if she was cooler, there'd be more of those bonds.
There's more energy in the system, and so those bonds are able to break and move around and exchange in the lattice with each other. So the thing that is really cool about this technique is since, for example, a dinosaur eggshell grows within the mother dinosaur, the temperature at which that calcite mineral shell of the egg is growing is the internal body temperature of the mother dinosaur.
And recent work has shown that it's not just the dinosaurs that evolved into birds that were warm-blooded. Other, more distantly related dinosaurs were as well. But what does that mean for what we know of dinosaur evolution?
Dinosaurs sit at this interesting evolutionary point between living reptiles and living birds. And reptiles are cold-blooded and birds are warm-blooded. And so this question about what dinosaurs were, extinct dinosaurs, I think people have been wondering for a while, and there have been various attempts to try to get at this. What's really fascinating is that the dinosaurs we've studied, which represent all these major groups, we've actually found that all of them had body temperatures warmer than their environment, which suggests that they had capabilities to use their metabolism to raise their body temperatures above the ambient. And so if all major groups of dinosaurs have this, what it suggests, what the most reasonable assumption then is that this is really something they acquired early on in their evolution. It's an ancestral trait if it's representative of the whole group.
Robin Dawson speaking with Adam Murphy there, and that work that she did while she was at Yale University has just been published in the journal Science Advances.
Finally, the British government has just granted its communications watchdog Ofcom sweeping new powers to police social media. The announcement is linked to the death of teenager Molly Russell, who took her own life in 2017 and whose Instagram feed was later found to contain graphic suicide-related content.
Ofcom are now going to target violence, cyberbullying and child abuse. But how effective can they actually be? Well, with us is Peter Cowley, our regular tech correspondent and local angel investor. So, Peter, first of all, what are Ofcom going to do?
Before we do that, can we just a bit of background? So what we have here is a spectrum between clamping down completely, therefore removing the possibility of the tragic case of Molly Russell, and full freedom of speech. Now, we know it's not a good idea to clamp down completely, but we know at the other end there's positive benefits for being able to communicate. This has been a problem for some time now, many years, and the government published a white paper called Online Harms last year. They're putting together some rules, and at the moment they're saying things like it's got to be proportionate and risk-based, and it's got to rely on the platforms to self-police, etc.
How enforceable is this going to be, though? Because the issue is that if I'm a tech company and I am hanging out in the back of Beyond somewhere and I've got servers which are not located in the UK, I don't really care what Ofcom say.
Yeah, well, the German system has fines of up to 50 million euros if you get it wrong. I don't think that's been invoked yet. But there's various things they can do. They can switch off the domain at the ISP.
But can they, Peter? Because the thing is, the domains are registered not in the UK, some of them, are they? Some of these domains are actually held in servers in America. So you'd have to have some kind of bilateral relationship with the Americans and say, I want to turn off Facebook. And then America might have antibodies about that and say, no, you're not doing it.
Yeah, that's not quite true because China manages it, doesn't it, with 10,000 plus domains.
But China has this sort of giant firewall infrastructure, and Russia are talking about having one as well, aren't they? Where basically there's a ring fence on the internet around a territory or geography, and they control and potentially probably inspect everything that's going backwards and forwards across that firewall, so they could just turn off a domain.
Yeah.
I mean, are we talking about, therefore, the internet was this amazing free-for-all for many years? Are we now talking about carving it up?
Exactly. I don't know. I think the most likely thing that's going to have some force is to have a named individual who is responsible within an organisation. Who wants to put their hand up within, say, Facebook UK and say, me, because this is an imprisonable offence, effectively. That will then trigger some change internally, because in the end, switching off the domain, as you say, is too extreme.
Well, let's say Ofcom can enforce these rules. From the companies then, say Instagram or Facebook, to what extent are they already set up such that when you look at a certain type of content, the algorithms then give you stuff that's very related to the stuff you're looking at? They say, oh, you like this, we're going to give you more of that. To what extent are, if they're trying to regulate certain types of content, they fighting against their own internal...
Exactly. So I'll give you an example in a minute. But what you've got to look at is the problem that we don't know whether they're content providers or just platform the content from other people. They clearly are editing content coming. I mean, I don't know if you noticed, but today Mark Zuckerberg in Switzerland actually said something. He said they have 35,000 people working on this problem. It's costing $5 billion a year. They're switching off 1 million accounts a day at the moment. So they're obviously trying to do something, but clearly, how can they do enough?
Presumably that's one of the ways in which this problem can be solved, because algorithms can be written to spot the very things that enable these sites to bring people together who have common interests and beliefs.
Yes.
That whole technology could be turned to find the very things that we decide are not in an interest of an individual or society.
It's already been done. There's no way that 1 million accounts can be switched off by 35,000 people. They can't read it all. And even if they do read it, how do you interpret it as a human being?
The problem is, and I've run into this, where Google has decided that certain pages on The Naked Scientist contain graphic content. And actually, when you look at it, because we're a medical site, we've got pictures of bits of the body.
Flesh,
Now they're not necessarily naughty bits of the body. There was one page the other day that Google had condemned, and it was a wound being sutured. It was very informative, but they decided the image there was not in the interest of people to see. And in fact, it was fundamental to explaining how the technology...
That's freedom of speech again, isn't it?
But that's what I'm saying. I think there is a danger we're going to take the wrong things down with it.
The three of us in this studio all have our own views about this, and all your listeners will have their own views. And who makes that decision? How can we expect the government, in whatever form, to get it right? We can't. But they must do something to prevent the sort of things that are causing harm and suicides.
Peter, thank you very much. I guess that's one which we'll have to return to. Thanks very much, Peter Cowley.
And if you'd like to find out more about the news stories we've discussed, the links to each of the referenced papers are on our website, www.nakedscientists.com, along with the transcripts of every interview for every Naked Scientist show. And why not follow us on social media? We're at Naked Scientists, and feel free to leave us a review. We love to hear from you wherever you get your podcasts.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
Now, in the second half of our programme, Chris investigates a type of cancer that unfortunately is all too common.
Every year, about 50,000 men are diagnosed with prostate cancer in the UK. That's about a quarter of all male cancer diagnoses in that period. It sounds alarming, doesn't it? But at the same time, people often say that this is something that you die with rather than of. So what is the bottom line? My name's Dr. Chris Smith, and in this programme I'm going to find out how prostate cancer presents in the first place, what it looks like down a microscope, and how we diagnose it. But first, who's most at risk of this? Vincent Yanapragrasam is a urologist specialising in this topic at the University of Cambridge.
Everyone is at risk. And they used to say that if you live to 80, then 80% of men will get prostate cancer, and that's probably true. Having said that, certain groups are at higher risk, so particularly men of African descent have a higher risk, or if you've inherited certain gene mutations, in particular the BRCA mutation, but that's only about 1% or so. And we still don't quite understand why some men develop prostate cancer more rapidly than others do. But what we do know is that when a man presents with prostate cancer, the older they are, the more likely are they to have more advanced disease. So really, prostate cancer is a disease that evolves over time. And the real challenge here is to not overdiagnose cancer which is early, but at the same time not picking it up when it's too late.
And that's what we're going to explore as we go through the programme. But first, let's back up very slightly. What is this gland that we're discussing? Well, the prostate's found only in men. It sits below the bladder, surrounding the urethra, the tube that drains urine. Normally, the prostate's about the size of a small satsuma, and its job is to produce many of the components of seminal fluid. As a man gets older, his prostate naturally enlarges, which can obstruct the flow of urine. And although this enlargement usually has nothing to do with the development of cancer, it's nevertheless often the trigger that sends a person to see their GP, who then investigates further.
My name is Vinitha Thangappan. I am Macmillan Lead Nurse for Urology and Prostate Cancer Specialist Nurse at Addenbrooke's. Usually the urinary symptoms are caused by the enlargement of the prostate. All men have their prostate getting enlarged from their 40 years of age, and when it gets bigger, it causes obstruction to the water pipe because the prostate sits under bladder and the water pipe go through it, and when it gets enlarged, it squeezes the water pipe, which causes a restriction to pass urine, which then lead to the symptoms like getting up at night or frequency or urgency like that. The enlargement of the prostate is majority of the time due to benign enlargement, but sometimes advanced prostate cancer can cause some of those symptoms as well.
Which is why if someone has them, it's important that they should get checked.
Yes.
Are there any other symptoms? Because we've dwelled on urinary symptoms. Are there any other ways in which prostate cancer could present in the first instance that the person might not think it's got anything to do with their urinary system?
The other symptoms is usually bone pain, and that's majority of the time when it is an advanced prostate cancer. Usually patients get bone pain and they put it into their stretched or done something physical to their back or things like that.
So when the person goes to see the GP with these symptoms, what sorts of things would they expect their GP to have done or checked or have put in place before they would send them to you?
So usually if patient presented to the GP should take their history, especially the urinary symptoms when it started. If it was a long-term urinary symptoms and getting worse, it's usually because of the enlargement of the prostate. But if it was sort of happened in short period of time, could be urinary infection. And so it is important that GP checks a urine sample, and then if there is no infection, GP should do the blood test, prostate-specific antigen, and usually the GP will explain what is the prostate-specific antigen, what can make it go high, and things like that. So that's what I'm expecting the GP to explain to the patient before they make that referral.
So significant emphasis is placed on the PSA test. But what actually is prostate-specific antigen, and how does it highlight the possible presence of prostate cancer?
It's a protein secreted by the prostate gland in its natural functions. It just so happens that prostate cancer cells selectively secrete more of it. It's not a perfect marker, and its accuracy is maybe about 60 to 70%, but certainly the higher it is, the more likely it indicates prostate cancer.
When you say the accuracy is 60 to 70%, what does that mean? Accuracy for what?
The accuracy that it predicts the presence of a prostate cancer. Now, it's very important to remember that the bigger your prostate is, the more PSA you produce. So, in fact, if you naturally have a large prostate, you're going to have a high PSA.
How does it get from the prostate and into the blood?
It's secreted in the bloodstream from the prostate gland.
So a person who has a tumour growing, the reason it's higher is what? Because more of it's leaking into the blood, or because the cancer is actively making it?
Yes, exactly right. The type of cells that produce prostate cancer are known as the epithelial cells. And the cells of origin from prostate cancer tend to arise from this compartment, and that's why they tend to produce more PSA.
So the GP gets a PSA test result. Presumably they don't just act on a one-off. If they saw a value that might be of concern, they'd repeat it before they did anything else.
You would hope so. And the reason that's important is that the PSA can be affected by other things. Sexual activity and ejaculation will elevate the PSA, reflecting the function of the prostate. It's an ejaculatory gland that produces secretory substances. And the message that we try to give out is always to repeat the PSA, because a single high level may not be indicative. Having said that, we would always take any PSA elevation seriously for at least assessment.
So how do we interpret the results of a PSA test then, given that it can be raised for many reasons other than just cancer?
It's a challenging issue. And so this idea of age reference PSAs have come about. Now that means that you have brackets of age where the PSA is allowed to be higher. So it's got to do with the natural enlargement of the prostate and an assumption that if you're in a higher age bracket, you've got a bigger prostate. So these age reference PSAs are used as guides in screening programs where they look for prostate cancer. They've set baseline cutoffs, like, for example, a level of 3. But of course, the PSA can vary from 0.1 up to 4 or 5 or even 6 or 7 or 8 or 10, depending on the size of the prostate, and not have prostate cancer. So interpretation is a difficult thing, but primarily to do with what you would expect to be out of the norm for someone whose prostate is naturally enlarging.
And if that is the case, under what circumstances then should someone be referred to a specialist prostate clinic?
2 elevated PSA in 4 weeks apart, that is the referral criteria. Or if it is 1 high PSA and if the examination of the prostate is abnormal, then the GP should refer to secondary care on a 2-week wait basis. So that's a cancer pathway basis.
In other words, it's cancer until proven otherwise, and that person's coming your way, or at least to someone like you.
Yes, that's correct. So usually, majority of the hospital, there is a triaging system where either doctor or the specialist nurses look at the referrals every day and then direct into the right pathway.
And that system will look at this individual and check things like, make sure there's no other reasons like a urine infection which may have caused this. But if it's looking as though the PSA is elevated for age, then classically now, the patient will be invited for an MRI scan first to see if we can pick up anything or see anything.
MRI stands for magnetic resonance imaging. It's a non-invasive scanning technique that uses a powerful magnetic field to enable doctors to see the body's internal structure in 3 dimensions, meaning they can measure the size and shape of the prostate with great accuracy, and even see inside it and other tissues.
What the MRI can do is it gives you a value of how big the prostate is. Because I mentioned to you that if your prostate is large and your PSA is raised, it could just be that's normal for you. So in fact, correcting the PSA for the size of the prostate, something we call the PSA density, is actually a much better test. And here's an interesting thing, that if we were able to transfer that to primary care, and then actually we might be able to give GPs more confidence about referring or not referring. And that's something we're looking into.
What proportion of prostate cancers are detectable straight away on an MRI? And what fraction do you subsequently think, well, it looks negative, you do a biopsy and then find they do actually still nevertheless have a prostate cancer?
So these are very complex questions. And in fact, MRI is part of the diagnostic pathway now, but many issues still remain, because in the end, unlike PSA, which is a blood test you measure in a lab, the MRI interpretation is down to the experience of the radiologist. And if they are a very good radiologist, the ability to see things is much better. But if you're not, then it may not be so good. So everything is always a balance of risk and benefit. With regards to the MRI, they have come up with a scoring system known as the PIRAD system. And this PIRAD system gives a score of between 1 and 5 in terms of how certain they are that there might be something. And if you have a lesion score, say 4 or 5, that's pretty sure that it looks abnormal. That doesn't mean it's cancer, but it looks abnormal. And the chances are that it will be a cancer when you biopsy it. Scores of 1 and 2 are almost negative, and so in that case it's probably unlikely that they can see anything. That doesn't mean there's no cancer there, but it's unlikely. Where the situation gets a bit more murkier is around the level of 3, because this is a bit of uncertainty. Now, in general terms, if you were to biopsy men across that scale of 1 to 5, your likelihood of finding prostate cancer gets higher with each level. And the other thing it does is it tells you where these abnormal areas are, so you can actually target your biopsy to those areas.
MRI scans can therefore help us to highlight areas of interest and predict the risk of finding a cancer. But the diagnostic gold standard is a transrectal biopsy. This is where, guided by an ultrasound probe introduced into the rectum, small pieces of tissue are taken from the prostate, including from areas that are judged to be both normal and those that might be cancerous.
Transrectal biopsies are done in outpatients in our clinic, and we have special machine that sort of help us to outline the MRI, and then target is outlined, and then ultrasound helps the consult or the doctor to target those areas. So it's targeted biopsy, which is done through back passage if it is that is what patient need.
So you use the scan to inform, combined with ultrasound on that day, where you want to sample. So you make sure you basically are sampling the bits that you spotted in the scan were interesting.
Yeah. So we do targeted biopsy, which means we take 3 or 4 samples from the target, which is the suspicious area within the prostate, and we do standard biopsies like 6 from the left side of the prostate and 6 from the right side of the prostate. So the reason for that is majority of the time prostate cancer develop in the peripheral area. So to make sure we are not missing anything, we do target plus standard.
What's actually involved in doing that biopsy?
What they are expected to do is lie on their left-hand side. And then usually a probe go through their back passage. The probe itself is slightly bigger than the thumb, and that is all fitted with equipment which guides the needle. Before the biopsy, we numb the prostate gland to make sure we can take the sample pain-free. Patients will feel a scratch like you are at the dentist, but it's wrong end. After numbing the prostate, they take the sample. What I have heard from patients are they feel a heavy touch-like feeling when they fire the biopsy gun. That can be uncomfortable.
So it's basically firing a needle through the gland on a trajectory that you've chosen to sample the right bits. And as it fires the needle through, it's grabbing a core, as it were, of that tissue and pulling it back for you to then send to the pathologist.
Yes, that is right.
What side effects will the patients experience afterwards, or what do you warn people when you go home after this? This is what to expect.
Main side effects is bleeding through back passage and blood in urine. Usually we tell patients make sure they drink plenty to flush their system. The other side effect is blood in semen, which can last sometimes 4 to 5 weeks, and it doesn't present as bright red blood in semen. It usually blackish old blood, so we do say don't get alarmed, it is something normal to see. The other side effects is infection. So there is a risk of urine infection, and there is a risk of septicemia, which is infection in the bloodstream. So we do give antibiotic for 3 days post-procedure.
Increasingly, though, practices are changing, and it's now possible to make better use of the scan data to achieve even more accurate sampling from the prostate.
And so we are now starting to do much more what we call transperineal biopsies. And this is where we still have a probe in the back passage, but the needles themselves pass through the perineum, the space between the rectum and the scrotum. And there are many ways to do that. The classical way is to do it under general anesthetic in theatre, but that's expensive. But you can get to the front and the sides of the prostate much better than you can from the transrectal approach. But we and others are starting to do local anesthetic transperineal biopsies using new devices that have been developed, and we have one in Cambridge which is going to be very cheap, and we think will revolutionize how the biopsies are done, because you're doing it through the perineum, but also under local anesthetic, and about for the same time as well. But in essence, all these biopsy methods are designed to do one thing: to take samples from the prostate to make a diagnosis. Now, we used to do what's called systematic biopsies, which means basically taking samples from different sectors of the prostate. Now we do that, but also targeted biopsies, which means going to the area the MRI has defined. And that is the samples we take and then send off to the laboratory for our histopathologists to look at and tell us what they find.
The biopsy samples are small cylinders of tissue collected at each of the biopsy sites in the prostate gland. These are sent to the laboratory where pathologists like Michael Eden look carefully at each of them for the telltale signs of cancer.
My role as a consultant histopathologist is to diagnose prostate cancer after a surgeon has taken a sample of tissue from a patient that they suspect has prostate cancer.
And when you receive these specimens, how do you investigate them?
We would take each of those bits of tissue. That tissue is cut into very, very thin sections because the cylinder of tissue contains thousands and thousands of cells, but actually down the microscope what we want to see is 1 or 2 cells at a time. So we will cut very, very thin sections and we can then stain it and then look at those cells which have been stained down the microscope.
So a lot of your diagnosis is literally made by eye. You look down a microscope and you're looking for cells that don't quite look right, or arrangements of cells, how they're organised inside the prostate that doesn't quite look right.
That's absolutely correct. So depending on the cancer, we'll be looking at what do the cells as individuals look like? What are they doing as a group? Are they growing together in a way that looks abnormal?
What can you say about the likely behaviour of the cancer based on the appearance when you see it? Can you give some kind of insights into how likely it is to progress or grow aggressively or be a more indolent disease?
Yeah. There are a number of things that we can look at to try and give an indication to the surgeon and to the patient as to how that cancer will behave. The first thing we try to work out is what type of cancer are we dealing with. The second would be to assign what's known in histopathology as the grade of the cancer. Does it look very, very aggressive, in which case we think that this cancer is going to behave very badly, or do we think that the grade is very, very low, in which case we think it's behaving more like normal cells, so it's less worrying. The third thing we look at is, do we have any evidence to show how far the cancer has spread? By that I mean, has it spread into other parts of the body?
You've got a microscope sitting here.
I do, yeah.
Could you show me what the specimens you look at look like?
Yeah, absolutely. So I've got a selection of prostate biopsies here. Typically, prostate cancers are graded using a Gleason score, and the Gleason score typically comprises 2 numbers which are added together, and the numbers can range from 1 to 5. We don't usually see grade 1 and grade 2, so typically we're looking at a number ranging from 3 to 5, added to another number ranging from 3 to 5, and that gives you a total number which will range from 6, which would be the lowest, up to 10, which would be the highest or the worst grade. So what I'll show you now is a prostate cancer, which is Gleason 3 plus 3, which equals 6, which is the lowest grade. So as you can see here, these sac-like spaces are lined by 2 layers of cells. There is what's called an epithelial layer of cells, which produces secretions in the prostate, and underneath them a myoepithelial layer of cells, which are smooth muscle in origin. If we see these smooth muscle layer of cells, I can be fairly confident that this is probably benign.
So just to describe what I'm seeing here, we've got this thin strip of tissue and there are lots of little holes. This is because the prostate is a gland, isn't it? So it's going to have these structures which you described as sac-like, which is where the secretions are going to be produced and then put into and then expelled from the prostate. And around those sac-like areas are rows of cells which have got nice purple centres. Those are the nuclei in the middle of the cells, aren't they? And they're all very nicely organised. And underneath those, you're saying, and I can see them clearly, there's another layer of cells.
That's right.
Those are the myoepithelial cells that you're saying, if they're there, it's usually a sign of good health.
Correct. That's absolutely right. So my job is to look through systematically, looking to see really, is there anything in the architecture of the cells, so how the cells are arranged in relation to one another, that would indicate this might be prostate cancer rather than benign prostate glands. And one thing that does catch my eye, which you get with many years of training, is that areas of prostate cancer architecturally show glands which are quite closely spaced together, and they look a bit smaller.
This bit over here that you've now highlighted, and I see you've drawn with red pen on the slide, so that you must have been the hot spot that you noticed. Yeah. That's very compacted and squeezed together. It's subtle, though.
As you zoom in on them, you'll see the 2 main features, really. One is that the myoepithelial cell layer is missing from many of these glands, if not all of them. And the nucleus of the cell, in some of them you can see a little dot, which we would call a nucleolus, which is quite prominent at this magnification, which you don't see in normal prostate cells. So that, combined with the absence of myoepithelial cells, suggests to me that this is prostate cancer. And then at that point, if I'm happy it's prostate cancer, then I would be looking at trying to grade it to see how aggressive it is.
Preparing the tissue and then waiting while pathologists like Michael study it and report the findings takes a week or two. When the results are ready, the entire clinical team, including the pathologists, radiologists, surgeons and nurses, then meet to discuss each patient and the best way to manage their case.
It depends on the biopsy type, because if it is transperineal biopsy, we take more samples, so that take longer, usually 2 weeks. But if it is transrectal standard biopsy, it usually take 10 days. And then we discuss the case in our multidisciplinary team meeting, and we see the patient and give the results.
And at that stage, what do you say to the patients, or what information can you impart to them, and what's the next step?
So in the result clinic, we will have the information about the biopsy result, whether it is a cancer or not. If it is a cancer, what grade of the cancer, and we would have discussed what treatment option is available for patients. So when we see the patient, we will discuss the findings of the biopsy, say advanced or is it locally advanced or early prostate cancer, and we discuss the treatment options with the patient.
We can now work out with quite good accuracy what the likely prognosis, meaning to say the outcome is, with or without treatment, and that's what we use together with the patient to work out what to do next. The first thing to know is that there is absolutely no evidence that rushing through a diagnosis of prostate cancer has any impact on survival. So it doesn't really matter how long it takes. The key thing is that it's done well. In general terms, from the minute somebody presents to, say, the hospital for investigations, to get an MRI scan, to be seen, to have a biopsy and get a diagnosis, we try to do that within 2 or 3 weeks. And then we can make decisions about what to do next.
Some people listening to this may be wondering why it is that we have screening programs for certain diseases, but we don't have one for this, given that you've said that 80% of men aged 80 may well have prostate cancer, which argues it perhaps would be worth looking for. It's therefore a very common condition. Why is there no screening program for prostate cancer?
So the problem with screening in prostate cancer is not the fact that it doesn't save lives, because it does. The problem is that it picks up a lot of disease which never needs treatment. And that overdiagnosis can also lead to overtreatment because of poor understanding of the natural history of disease, a sort of instinct to treat rather than monitor. And if you look at the statistics as well, it's very hard to justify it. So, for example, in prostate cancer terms, the lifetime risk of developing prostate cancer is nearly 10 times the risk of dying of it. You have to find an awful lot of men to save a single life. But the problem with screening is that it's tended to rely on a single test at a single point in time. And the test isn't good. PSA, like I said, is a nice test, but it's just not good enough. And so a lot of our work, a lot of work of others, is actually trying to do two things. Define what is the right time point to pick it up, and secondly, trying to get a better test to pick it up.
Are you having any luck?
Yes. There are actually a number of tests out there which are already better than the PSA. The problem is that they cost more as well. We are very interested in actually combining better tests with a much more evolving way of looking at this, because the way we see it is that each man's risk changes with time, and how we put that two together is what we're exploring at the moment. But I do think we have the tools to be able to better detect cancers which are going to spread, for example, and actually intervene early enough.
And would that translate then into a saving?
And that's exactly what the Holy Grail is. Because in the end of the day, screening or early detection for cancer actually has a converse side, which is not picking up things that you don't want to pick up. And that in itself is a goal which I think is worth doing. Because if you can reduce the number of people you're going to be looking for something, that means you can get your resources redirected to finding the ones which are important. Most of the time, we are so fixated on finding and more cancers. That's what we want to do. We forget about the fact that there's a lot of people who are investigated who turn out not to have cancers. And that is actually a bigger burden for the health economy, for the individual. And ultimately, if you do diagnose something which is too early, you do condemn someone to being monitored for something which they didn't know about. It's a little bit like saying, if we started to do genetic tests at birth for everyone, what it tells you is a potential that you might get something rather than you will get something. and then you're going to end up stressed about it for all your life, and it may or may not happen.
But obviously, for some people, it does happen.
My name is Michael Langford. I'm 88 and a half, and I'm a semi-retired professor of philosophy. 18 months ago, I was diagnosed with prostate cancer, and for a time I was really pretty sick. But a combination of steroids and hormone treatment seems to have made me a lot better, though at one point I had to go in just over a year ago for blood transfusions because my anemia got very serious. But at the moment, touch wood, I'm a lot better.
How did you first detect something was amiss?
Well, there's a bit of a history. 23 years ago, while I was in Canada, I had a flow problem with my urine, and I had a little operation which made the prostate much smaller. Then everything was fine until about 18 months ago, when I was diagnosed with a combination of polymyalgia and prostate cancer, which they then discovered had spread, the cancer had spread to my femur and my ribs.
Did the GP immediately suspect that it might be prostate cancer, or was it because we should be clear, a couple of years ago, I mean, you were doing martial arts. You're a very fit and active person.
I was, until 18 months ago. The doctor diagnosed the polymyalgia and wasn't sure what else was there, and I was sent for more blood tests to Addenbrooke's, and there they found that I had quite severe prostate cancer.
What test did you have to make the diagnosis and confirm what it was?
I was sent for an MRI scan, and that showed that I had secondary growth in the femur and in the ribs.
You didn't have any biopsies? It wasn't deemed necessary then?
I didn't have biopsies. They were quite sure I had cancer anyway.
And how did you take that?
I was a bit shaken, but being in my 80s and having friends who've got prostate problems, I knew the old saying, almost all old men die with prostate cancer, but not of it. And I took consolation from that. But a year ago, when I was feeling really down, I did begin to wonder whether I had to start rewriting my will and so on. My family were very supportive.
And what actually then happened to you? So you get the diagnosis. Obviously, you're saying it did hit you a bit hard, but clearly you've bounced back because you look much better now.
Well, I was the lowest about a year ago when I had to have blood transfusions because of the anemia. And then as soon as I started on the hormone treatment, I gradually began to feel better. And apart from one bad week last summer, which I think was an infection of some kind, I've been continually much better. I do have trouble if I walk more than a mile, I get out of breath. And if I go up more than 2 or 3 flights of stairs, I get out of breath. So I pace myself. And I still teach a bit and lecture a bit, but I try to take a lot of rest.
And what have they told you about the prognosis?
Well, they were quite honest about this. When I first was diagnosed 18 months ago, one of the oncologists said the average life expectancy for someone presenting as you did is about 3 years, but it can vary from 1 year to 20 years. You know, in other words, it's very, very dependent on how you respond to the treatment.
And how are they keeping an eye on you?
I'm called in to see the oncologist about every 2 or 3 months, and I have blood tests about once a month. And my anemia level I keep an eye on.
Do they rescan you?
I've had 2 scans. They're very expensive, so understandably I don't expect them too often. I had one about 3 weeks ago, which I haven't had the results of yet, but they haven't phoned me up, so I'm hoping they're good.
Well, you know what they say, ignorance is bliss, isn't it?
Yes. Yes, that's probably true.
And my thanks to Michael Langford for sharing his experience with me, and also to Michael Eden, Vinita Thangapanaya and Vincent Yanapragasam. And do keep an eye out on the feed for our landmark look next at the landscape of AI in healthcare. The Naked Scientist comes to you from Cambridge University and is supported by Rolls-Royce. I'm Chris Smith, thanks for listening, and until the next time, goodbye.